in my lab, we are interested in two aspects of selenium. And one is study the role of selenium at the nutrition level. We know at nutrition level we study the senoprotein. So that will affect the senoprotein expression. So the general idea is that with the functional senoprotein you can maintain the known stability. But in the cancer part is different. Our interest is at the supranuclear level. So at this level, you actually generate oxidative stress. Although some senoprotein is antioxidant, they can decrease ROS. But if you have the selenium higher than the nutrition, you won't change the senoprotein expression. So you have too much selenium, you actually generate ROS. And this actually causes stress on the genome. So this part is to protect against aging, and this part we propose that they protect against cancer. And in this talk today, I'll only focus on this cancer part. So selenium actually is a very old chemical to study aging. So actually this has been 100 years. The first case known is in 1911. There's a scientist. So he just injected a form of selenium, inorganic form called selenite, directly through the tumor in mice, and he can see the shrinkage of the tumor in the mice. So 100 years ago, there are already people to consider selenium as an anti-cancer chemical. And also later on, based on the geographic or the clinical studies, and they found there is a correlation between the selenium status. So the correlation between the selenium deficiency and the cancer incidence. So this is based on the geographic data or the clinical data. So the geographic data, that means they collected look at the individuals live on this land and look at their cancer incidence, and then also to see measure the selenium content on the land. So they found people who live on the land low in selenium, they have a high incidence of cancer. But this is a long time ago. That's when people they eat the food coming from the place they live. So nowadays this study is less possible because now we have food grown in far away, and we eat. Let's say you can eat food planted in Japan, in the United States. So, but in 40 years ago, the food sources is more limited. So the other thing I want to point out is this definitely is to study the toxicity of selenium, because you inject, you not only can shrink the tumor by injecting selenium, you can inject other things. You inject high dose of zinc, high dose of copper, you still can kill the tumor. It's just because you have the toxic amount of chemical in the tumor. And this is not our interest at this moment. Okay. So now it comes of two controversial or contradictory result of a big clinical trial. So one is first published 15 years ago. This is called nutrition prevention of cancer studies. So this is done in the United States, and provided oral dose of selenium-enriched yeast. And this dose of selenium is 2 to 3-fold higher than the nutritional need. So the nutritional need is about 50 to 70 micrograms a day, and they give the individuals with 200 micrograms of the selenium for 30 years, and they found reducing cancer incidence, including prostate, colon, and skin cancer. And what's in this selenium-enriched yeast? And 65–80% is one of the amino acids, cetanol and thymine, and there are also 20 other unidentified compounds. And then 2 years ago, there is another big clinical trial based on the NPC trial. So in this study, because the majority of the selenium compound in the yeast is selenium methionine. So they used only selenium methionine and specifically look at the prostate cancer. And they found the selenium methionine does not prevent prostate cancer. So there must be something different between these two clinical studies. And at least what I think is the chemical forms is different. This selenium methionine, at least based on this result, it shows that selenium methionine is not an active form to suppress prostate cancer. Okay. And also later on in 2009 and 2008, so in animal study, this group shows another form of methylselenium acid, but not selenium methionine, can suppress prostate regenesis in two mouse models of prostate cancer. So this is a complicated selenium metabolism in the body. So to make it short is if you have nutritional level of selenium, they will go through this pathway and to support selenium protein expression. But if you have too much selenium, and after this pathway is filled, and extra selenium, they will actually induce the formation of oxidative stress, hydrogen peroxide and peroxide. So when I get started, when I have an independent lab in 2007, so I look at the old publication, and I found at least 50 publications. They try to find out the reason why selenium can kill the cancer cell or fight against cancer. So I found what they did is use high dose selenium and then treat the cancer cell. And then they found the cancer cell get apoptosis, that's a programmed cell death. And also they have an induction of selenium. So all those publications, I think why selenium can prevent cancer is because selenium high dose induce apoptosis, kill the cancer cell. But in my view, if we want to convince the individual to eat the supine nutrition level of selenium and that can prevent cancer, we should mitigate the tumorigenesis at the early stage of the tumorigenesis. If you target the cancer cell that's too late, I don't know how you can say that prevention. It should be the treatment. Normal cell. Yeah. Yes. So my point is, if you really can treat cancer, you will kill the normal cell first. You will not have the selectivity. Well, the other thing I forgot to mention is, if you have any question, you can ask anytime. Okay. So in beginning from 2005, and in the DNA damage field, they begin to make a conclusion that the DNA damage response and the senescence, these are the two critical events that happen at early stage of tumorigenesis. And this group, they participate in some of the key findings. So in one of the review papers he published, so you can see this is the stage of tumorigenesis from normal cell to become precancerous and becoming a cancerous cell. So the green color means the proliferation index. So during the transition from normal to precancerous, the proliferation is decreased. And this together with the induction of the DNA damage response as well as the senescence response. And this event happened, a decrease when precancerous cells becoming a cancer cell. So in other words, the induction of senescence and DNA damage response, these two are critical events happen at early stage of tumorigenesis from normal to precancerous stage. So in case you don't know, the senescence, this is a permanent withdrawal from the cell cycle And this is also known as the cell age. That means if the cells are entering the senescence, they cannot enter the cell cycle. And this is bad for longevity. But this, in cancer, this is a way to stop the cancer cell to move on to the next stage. So senescence as an early event of tumorigenesis, and this is being identified in one of the key landmark applications. So this MRC5 is long normal cells. So if you inspect the normal cells with the mouse, this is oncogene, and they're quickly undergoing the senescence. So that is easy assay. You can measure the beta-gal expression, and this is one important convenient marker of senescence. So once you have the oncogene induction in the normal cell, so this is like here. So if you express mouse oncogene at this transition, and you quickly see the increase in the senescence state. And this oncogene-induced senescence at the early stage, at this stage, require ATM, because if you suppress the expression of ATM, this is a DNA damage kinase in this cell, and if you induce with oncogene, you don't see the senescence. The blue color, that means senescence. So that means the oncogene-induced senescence at this early stage. And then this event requires ATM kinase. So ATM mutation can cause genetic disease. So if you have ATM mutation, there is a disease it's caused by ATM mutation. It's called ataxia telangiectasia. And this is due to the mutation of the ATM gene. And this is a genetic disease. And this is autosomal recessive. That means you need to have two alleles to be mutated. Then you will see the phenotype. You will see the ataxia telangiectasia. And these two words means ataxia means you have poor motor coordination. And usually one of the hallmark of ataxia telangiectasia is the cerebellum degeneration. And the other phenotype is the telangiectasia. That means they have a small dilated blood vessels on the skin or on the tissue such as in the eye. So this is the telangiectasia. So those with the ataxia telangiectasia, they usually have the problem to move motion coordination problem, as well as those dilated blood vessels. And they also are prone to lymphoma as well, and also they are immunodeficient. So DNA damage response. In response to DNA damage, especially double-strand break, we have the sensors to sense the DNA double-strand break. And then, okay, this is ATM. This is the gene responsible for the ataxia telangiectasia. And this is a DNA PKC, is another protein in the same family of the DNA damage response. So once they receive the signals from the sensor protein, so they will be activated. And then what I do is to phosphorylate their substrate, the affected proteins, and result in the DNA damage repair, checkpoint activation, or even the senescence response. And this also includes to mark the site of DNA breaks. And when they are activated, they can phosphorylate into an X protein to form the gamma extract. And this is also a common marker of DNA breaks. So we know the mutation of ATM causes ataxia telangiectasia, but right now there is no known genetic disease associated with the DNA PKC mutation. So based on this, the selenium and the cancer field, as well as the marker or the early event of tumorigenesis. So a few years ago, we made a hypothesis that if selenium can counteract tumorigenesis, so selenium should activate the early barriers of tumorigenesis. That means the ATM-dependent pathway as well as the cellular senescence. So for the first experiment, we use different forms of selenium compound, organic form or inorganic forms. And because the supranutritional level of selenium induce oxidative stress, so as a control, in some experiments we treated with antioxidant.